Hello, everyone. My name is Ernie Guzman. I'm the senior manager of technical support at Twist Bioscience, and I'd like to welcome all of you to today's presentation entitled Cellular Reprogramming for the Scalable Production of Human Cells, presented by Dr. Michael D'Angelo, senior scientist at BitBio. Just a couple of quick housekeeping rules to cover. First is that all lines will be muted during the webinar, and this is to reduce any distractions during the presentation. Secondly, we will have a Q&A session at the end. Please submit your questions in the Q&A box. We will answer your questions after the presentation is complete. And finally, following the presentation, there will be a short 1-minute survey that we would really appreciate you taking. Your feedback will help us improve all future webinars. And now, I'd like to introduce our speaker. Today's speaker is Dr. Michael D'Angelo. Michael is a molecular biologist and genome engineer. He leads the BitBio team conducting technology development and gene editing projects, which are at the core of all of BitBio's cellular reprogramming activities. And with that, Michael, the floor is yours.
Thank you so much for that introduction, Ernesto, and I'd like to thank Twist Bioscience for the invitation to speak today on behalf of BitBio. Today in this webinar, I'd like to tell you about the need for better human cells and the promise of cellular reprogramming. I'll tell you about BitBio's approach to making human cells and how Twist gene synthesis has helped us. And I'll finish off by showing you some data and showing you some of the products that we've made using our opti-ox technology. So why do researchers need human cells? Well, human biology is often different from the animal models and tumor cell line models used in preclinical drug testing. And as a result, many drugs often fail in human clinical trials because of a lack of efficacy or toxicity that was not appreciated using these non-human or non-physiological human cell models. So where are researchers going to get good human cell models for drug testing? Well, you might think that the perfect human cell model would be primary cells from donor tissue. However, these have some problems. They are of limited source because they need to come each time from tissue donations, and they often display highly variable biology because they need to be isolated from complex tissues, and they come from donors with different genetic backgrounds, adding heterogeneity and noise to the system. There's also classical tumor-derived cell line models. These have advantages because they're easy and simple to grow. However, they're transformed and they don't represent normal human biology. And then you've got pluripotent stem cell derived models, which in principle have the ability to provide an unlimited source for human cell types. At the moment, however, their bottleneck is really realizing their potential. And at the moment, all of these three sources suffer from a lack of consistency, scalability, maturity, and purity. So if I tell you a little bit more about iPS cells, so these are cells that have been reprogrammed from mature human cell types back into a pluripotent state so that they're very similar to embryonic stem cells in their ability to differentiate into all the three germ layers of the human body and in principle have the ability to become any cell type of the human body. And so they have advantages in that because depending on where you get that starting cell type from, they have the ability to represent different human genetic diversity. They are self-renewing and therefore they're inherently scalable. And they are now have really robust cell culture procedures in place, which means that they're amenable for genetic engineering. And so what we really want to use, however, is these cells on the right over here. So we've got a neuron, a white blood cells, and red blood cells, for example. We want to get to a mature cell type from these pluripotent stem cells. And the traditional protocols for doing these are called directed differentiation. And really with these protocols, what you're looking to do is mimic development in the dish. So you're putting generally growth factor combinations on the cells to mimic what happens during a regular developmental pathways via various intermediaries and precursor cell types to end up at the final mature cell type that you're interested in. However, these tend to suffer from having very complex protocols with very expensive and complex media that needs to be prepared, and therefore they struggle with scalability, Also, the maturity and the purity of the final cell type you end up with is not always particularly where you want it to be. And so synthetic biology provides us with a new paradigm for how to make mature cell types from pluripotent stem cells, and this is the idea of reprogramming. So now, instead of the cells having to interpret a complicated extrinsic signal, we're now giving them a strong, simple to interpret intrinsic signal by overexpressing transcription factors. And in this case, we're imposing the cell identity and transcriptional network upon the cells, and therefore speeding up the differentiation process. However, standard reprogramming methods lead to polyclonal heterogeneity and low scalability, and this is where our opti-ox technology at BitBio comes in, and we're able to now get cells at the end that have high purity, maturity, scalability, and consistency. So what are we all about at BitBio? So BitBio's mission is to code cells to advance the well-being of humanity, and to do so, we apply the principles of computation to biology. Our current focus is to develop a scalable technology platform capable of producing consistent batches of every human cell type, and to enable research and drug discovery to move on from less efficient models and work with the cells that are actually affected by human disease. And a scalable platform of consistent cells will also be the basis for a new generation of cell and tissue therapies. So at BitBio, we have two pillars on which we're based. So in order to reprogram cells, we need to identify the optimal transcription factor code that can reprogram cells from the pluripotent state into the mature cell type of interest. And to do that, we've built our BitBio discovery platform. And this is a platform that allows high throughput experimentation coupled with machine learning to determine the optimal transcription factor code of interest that we can then use to program a mature human cell type of interest. And then once we've identified the optimal transcription factors, they are then ready to be put into our opti-ox platform, which is the way by which we overexpress the transcription factors needed for cellular reprogramming into the new cell type of interest. And I'll go into detail about how the opti-ox platform works on the next few slides. But what this allows us is to have cells that now have very high consistency, very quick speed to maturity, high scalability. They're very easy to use and cost effective. So opti-ox stands for optimized overexpression. And so older methods, for example, using lentivirus non-specific integration methods, lead to high variability in reprogramming. And this is because each gene expression cassette will land in a different genome location, and therefore leading to differences in expression level of the transcription factors, and therefore differences in the outcome of the reprogramming. So initially, experiments were done using a targeted system, so into a genome safe harbor site, using an inducible system, so in this case, the doxycycline inducible system. However, when this was first tested, what was seen was low expression and silencing of the transgene, in this case, GFP. So you can see patchy and low expression in this iPS colony here. And so from this is where opti-ox was developed. And so in a seminal paper from our founder, Dr. Mark Kotter's academic lab, what they did was a simple trick. What they did was to split the two components of the doxycycline inducible system. So we have a constitutive expression of the RTTA transactivator protein from one genome safe harbor site. And then at a second genome safe harbor site, we now have the tetracycline response element, inducible promoter driving expression of the transgene of interest. And now what you see is strong, stable expression. Every cell in this iPS colony is now expressing GFP. And so opti-ox is able to overcome these limitations of silencing. So this is great because with the inducible system, what we have is in the absence of doxycycline, pluripotency is sustained, and so these cells are now self-renewing and inherently scalable. And now when we add doxycycline, we have deterministic induction of a new cellular identity. And so these are examples from this paper showing reprogramming into these 3 cell types of interest in very short time frames. And so this is what it looks like in action. This is a time-lapse image of cells differentiating into glutamatergic neurons. What you can see is that within just 3 or 4 days, they're already displaying neurite outgrowth, and then they become post-mitotic and they form a dense neural network within a week. So what's really exciting about this is the speed at which they're able to form such a dense neural network. This normally takes weeks with classic directed differentiation cultures, and also the homogeneity that you can see really every cell in that culture is reprogramming. They're changing morphology at the same time, and the resulting cell population is extremely pure. So how's twist gene synthesis helped us in our mission? So in order for us to get access to different transcription factors that we want to test with the opti-ox platform, we use twist gene synthesis. And so what we used to do was when we had a new transcription factor, we would get it synthesized in the twist cloning vector. Then they would send it to BitBio, where me and my team would then perform subcloning into our opti-ox vectors. However, now it's even better. We've completed custom vector onboarding. So they now have our targeting vectors there. And what we can do is whenever we want new transcription factors to be tested, we can order gene synthesis, and Twist will clone them directly into our opti-ox vectors and ship them back to us ready to use. So this is fantastic because it's fast and it frees up the time that my team would have spent at the bench doing the cloning. It's also inherently scalable because if we were to order a large number of transcription factors, well, the amount of time that we would take to subclone them would scale somewhat linearly with the number of factors that we ordered, whereas at Twist it takes roughly the same amount of time to get one transcription factor as it takes to get 50 because of their high throughput platform. And we're also really excited to take advantage of their new high yield DNA prep offering, which will hopefully get us access to transfection ready DNA. So we'll spend even less time at the molecular biology bench and more time doing exciting things, plugging these into iPS cells and making exciting new human cell models. So BitBio's cellular reprogramming is really making a new benchmark for human cell models due to the high purity, the speed of differentiation, the consistency, not only batch to batch, but really cell to cell in each vial, and also the scale at which we're able to do production. And this enables a number of applications for basic research, both normal and disease models, and we're really building up a great capacity for genetic engineering of disease models. And this is enabling for developmental and cell biology experiments. It's also enabling for drug discovery. So both normal and disease models can be used for high throughput drug screening, target discovery, and high throughput toxicology screening. An interesting application is in bioproduction, and actually our founder, Mark Cotter, is also a co-founder of a clean meat company using the opti-ox technology in cells derived from livestock animals to make a scalable source of lab-grown meat. And of course, a really exciting application for this would be cell therapy, whether it be immune cell therapy or other regenerative medicine applications for different cell types. So now I'd like to show you some data from some of our human cell models. And so these will be our iPS-derived neurons and myocytes. So first up is our fully launched product, which is the human ioneurons GLUT product. These are ready for experiments as early as 2 days post-revival, and they are, majority are glutamatergic neurons, something like 88%, and a smaller subpopulation of cholinergic neurons. So what you can see here is some immunofluorescence staining for pan-neuronal markers beta-3 tubulin and MAP2, and then in orange here are the two glutamate transporters, which are specific for glutamatergic neurons, vGlut1 and vGlut2. Bulk RNA-seq analysis has shown that the neurons have a rostral CNS identity. They exhibit robust neurite outgrowth and form functional neural networks in as little as 17 days that demonstrate drug response. So what do our customers get? So at BitBio, the first phase here is performed, which is induction. So for 3 days, the cells are primed towards the neural fate, and then they're cryopreserved, and then you will receive one of these cryo vials over here with cells that are ready for plating and rapidly mature upon revival. And then it's a very simple process in the customer's hands. They go through a 4-day stabilization phase, during which doxycycline is kept in the media, and then a maintenance phase for however long you want to culture them, where doxycycline is now removed. And during that whole time, they've just got a simple glutamatergic neuron media, which is fully disclosed formulation, and this is enabling for our customers to customize it for their particular experiments. So we've done some deep characterization on these cells. So we've done single cell RNA sequencing. So what you can see here is a time course of reprogramming. So following anti-clockwise from the IPS state through some early time points here, 12, 24, 48, and then 72 and 96 hours, and then also some very mature cells over here, 2 and 3 weeks. And what you can see is the pan-neuronal marker MAPT starts to come on around that day 3 to 4 mark, and then is really expressed in every cell at the 2 to 3 week mark. And most of the cells are expressing this glutamatergic marker, with a smaller subpopulation that express this cholinergic neuron marker. And so our partners, Charles River, have used these cells in various different experiments. You can email us for a copy of this poster if you're interested at info@bit.bio. So what they've performed are some functional characterization using high-resolution multi-electrode arrays. So that is showing that the cells demonstrate electrophysiological activity. And so both at 2 weeks and then increasing at 3 weeks in culture, the firing rate, percentage of active electrodes, and the spike amplitude all increase. And also they have validated these for high throughput assays in 384 well plates. And so if you're interested, please email us and we can give you a copy of this poster. And now I'd like to talk to you about another cell model that we've created, which is the skeletal myocytes, so iomyocytes skeletal. This is in beta testing phase and will be hopefully launched soon. But if you're interested in being on the beta testing, being one of our beta testers, please get in touch. These are also ready for experiments as soon as 2 days post-revival, and contractility can be assessed as early as 3 days post-revival. They exhibit visible striated fibers and multinucleated myotube formation by day 10 in culture. And what you can see here is immunofluorescence staining, demonstrating robust expression of components of the contractile apparatus, including desmin, dystrophin, troponin, and titin. And also we validated by qPCR the expression of key markers, including various myosin heavy chain isoforms, as well as the transcription factor myogenin. And so here we have a video showing spontaneous contraction of these cells. So this is in the absence of any stimulation, 15 days post-revival. There they go. Always freaks me out when I see that down the microscope. Importantly, also these cells, if you add a stimulus such as acetylcholine, these cells contract very strongly then. So what I've shown you is data from our IoNeurons Glute fully launched product, and coming soon our iomyocytes skeletal. And also before the end of the year, we expect to launch our IoNeurons GABA, which is a GABAergic neuron product, as well as our ioglia oligo product, which is a human oligodendrocyte cell model. And if you're interested in those, please get in touch and we can let you know when they're going to launch. So at BitBio, we're developing the next generation of human cell models and providing the best cells for research and drug discovery. And our cells are used in research and disease modeling. They've also seen applications in 3D printing into 3D scaffolds. And so there was a recent paper out of Oxford University using our neuron cells in a 3D printed cell scaffolds, and also in high throughput screening. And at this point, I'd like to thank our partners, Charles River, who are using our cells in their high throughput screening applications, and Abcam, where our cells are available for purchase. If you want to go to the Abcam website, you can find the cells available there. And if you are an academic researcher or working in a not-for-profit organization, please make sure you take advantage of the discount code there so that you can get access to our cells for a really great price. So BitBio allows for the scalable and consistent generation of defined human cells. And the development of the BitBio lineage portfolio is driven by our discovery platform for the identification of novel and optimized transcription factors for new cell type reprogramming, and our opti-ox technology for the controllable expression of transcription factor combinations. And all of this results in cells that have high consistency, high speed to maturity, scalability, ease of use, they're cost effective, and they show really high purity. And with that, all that's left is to thank the team at BitBio that really make it a joy to come to work every day. The one problem with fun jumping pictures is that I think all you can see of me is maybe my shoe right there behind my boss who's blocking me, but it's a fun picture nonetheless. And yeah, thank you again to Twist for inviting me to give this webinar today. And with that, I think we'll take some questions.
Thank you, Michael, for your presentation. Really appreciate it. And now we'll take some questions from the attendees. As a reminder, if you have any questions, please make sure to ask them in the Q&A box. So a couple of questions here. How does the opti-ox improve reprogramming?
So reprogramming the cells is by directly activating a program that defines a particular cell identity by overexpressing a transcription factor from within. However, typical methods for overexpressing transcription factors leads to heterogeneous expression and therefore variability in the final cell outcome. Opti-ox gives a source where every cell is expressing the transcription factors at the same time and at the same level, and therefore what you end up with is a highly consistent reprogramming is a highly consistent reprogramming event.
Okay. Another question. So you've shown the power of the opti-ox to generate mature glutamatergic neurons and skeletal myocytes. What are the applications of your human cell models, and is BitBio developing any other cell types?
Yeah, so defined and consistent iPS-derived cells provide an excellent alternative to animal models and immortalized cell lines, which diversify during long-term culture. Our human cell models are used for research, for example, the study of neurological activity in the case of the eye neurons GLUT. They're also used in disease modeling and 3D printing. Because BitBio cells are consistent and scalable, they provide a good model for high throughput screening, and a great example of this is our partner Charles River, who have adopted our iPS-derived cells into their high throughput screening workflows for use in target discovery, validation, and screening services. And our aim is to democratize access to high-quality human cell models and provide researchers across academia and industry with reliable models for research and drug development. And so we're working on a wide variety of different cell types. So we will be releasing, as I mentioned earlier, some cell types in the central nervous system space. And if you're interested in trying them sooner as a beta tester, then please get in touch with us.
Okay. Another question is, how are you choosing which transcription factors to overexpress?
Yeah, sure. So that really comes down to the power of our discovery platform. And so that allows us to really do experiments in cells at the bench to screen high numbers and combinations of transcription factors and to determine the ideal transcription factor code that can make the final cell type of interest that we're interested in. And this is really powerful, and then the data coming out of that is coupled with machine learning methods which will allow us to predict these in the future.
Okay, another question is, are all the cells you provide diploid?
The cells that we provide, the cells that go into our manufacturing processes are diploid. So iPS cells are diploid cells with a normal carrier type. We do, that's part of our quality controls that we have at BitBio, and I believe we also check our cells at the end, but we'll need to double check with our product specialists.
Okay. How many transcription factors can you overexpress at the same time?
So that's a good question. And it also, I guess, comes back to how many transcription factors do you really need to make a particular lineage. At the moment, with relative ease, we can express up to probably 5 or 6 transcription factors. Of course, there are lots of ways that we can expand that out, and we're looking into that at the moment.
You sort of brought this up earlier, but another question is, how do you QC that you get the right program cells?
Of course, yeah. So there's lots of different ways that you can look at and benchmark the cells that you're making. One of the best methods, which I showed some data from, is single-cell RNA-seq analysis, because that allows you to look not just at a bulk measurement of expression of a particular marker, but it really lets you look, is every single cell in that population expressing that marker and to the same level? And it really gives you high order information about really what is the transcriptome of each individual cell and how well does that match the cell type that you're trying to model. Of course, the questions of what's the best benchmark is a little bit of a difficult one, because even primary cells, freshly isolated versus primary cells kept in culture, will display transcriptional differences. So that can make it a little bit difficult to say exactly what what the goal you're trying to hit is. But in general, that's a really good way of QCing. And then once you know classical markers that they should be expressing, then they can of course be tested either at the protein level by immunofluorescence staining, or at the transcriptional level by quantitative reverse transcription PCR.
Okay. Another question. Do you try to engineer the expression level of the transcription factors, or is it more of an overexpression versus not?
Sorry, could you repeat that? I just missed that one.
Sorry. So do you try to engineer the expression level of the transcription factors, or is it more just sort of an overexpression versus not expressed?
So, I mean, the beauty of the doxycycline inducible system is that it's inherently titratable. So if, like all of our products that we're releasing, we have established the correct concentration that is required to reach the reprogramming that we need. So that's built into the opti-ox system because of the dox-inducible system. So we have the ability to vary the expression level.
Have you ever made direct comparisons between the iPS cells produced and versus native cells?
Sorry, did you say versus native cells?
Yes.
I believe so. You don't mean iPS cells. You mean the final reprogrammed cell product. And yeah, I believe there was some slides in a recent webinar from our CEO that looked at ways of comparing the identity of the cells that we make in a reprogramming versus the mature human cell type that we're trying to model. So we have done benchmarking experiments like that. I don't have the data in this slide deck though. If you're interested, again, you can get in contact, or you can check out that webinar, which I think is accessible on our website.
Okay. Does your platform alleviate the need for growth factors during the differentiation process?
Yeah, that's a really good question. What I would say is that it certainly allows the simplification of the extrinsic signals, so the media components. However, at the moment, certainly there is still a need for some extrinsic cues. There still needs to be media in which the final cell types are happy and performing their functions. And so there are still, depending on the cell lineage, some growth factors or various additives in the media, but typically they're simpler than the media required for direct differentiation protocols.
The question is, the opti-ox integrating or non-integrating?
Yes, so as I mentioned, it involves targeted integration into genome safe harbor sites. So these are sites in the genome that have been widely used. They have the advantages that it's established that they are transcribed in the majority of different human cell lineages, and therefore they should remain open no matter which cell type you try to make. And yes, it is targeted integration at 2 different genome safe harbor sites for opti-ox.
Okay. Another question that comes in. Have you considered making disease models using these cell lines for neurodegenerative diseases? The question is, I do see a need for this in our work at our research foundation, and it is very hard to come by good human cell models.
Yeah, definitely this is something that we are pursuing at the moment, something that we're looking to expand. And if you're interested in a particular model, I urge you to get in touch with us and we can start a conversation. That is something that I think is a really exciting application of our cellular models. There's a lot that can be done with kind of wild type human cell models that are reproducible and consistent. However, it's certainly also very powerful to be able to do disease models with these. So it's something that we are actively exploring at the moment. So please get in touch if you're, and we can discuss what you're interested in, exactly what model.
Okay. Have you discovered many novel DNA sequences that can cause differentiation in iPS cells?
I'm not quite sure. I'm not quite sure what that question's asking in terms of novel DNA sequences. I mean, if we assume we're talking about novel transcription factor codes, I would say that our discovery platform is capable and we've shown that we've demonstrated that it's able to generate new transcription factor codes that haven't been published before. So we are definitely generating and discovering new transcription factor codes at BitBio.
Okay. Can you comment on the difference between your system and using an episomal plasmid to avoid any genetic changes?
So an episomal plasmid, so the main differences are going to be in the scalability and the homogeneity. When you introduce an episomal plasmid, you need to transfect the cells, which means that inherently that's difficult to scale. There are high throughput platforms for that now, but they're not widely available or even those are somewhat limited in their scale. So you need to transfect the cells. Then you've got issues where maybe there's transfection efficiency differences. So already you might have some cells that don't receive the episomal plasmid and therefore don't reprogram, or you need to use some sort of drug selection to enrich. And then you've also got the issue where you can't easily control the copy number of that episomal vector, and therefore you can lead to differences in the reprogramming outcome, both cell to cell in that batch that you've just tried to create, and also experiment to experiment, because every time you do that transfection, it's going to be a slightly different outcome. And therefore, for some applications, maybe that's okay, but for things where you really need consistency and purity, what you want to have is, every cell in the dish doing the same thing at the same time. And for things like high throughput drug screening, this is imperative, because if you have any extra sources of noise in your system, they can destroy your signal, and therefore suddenly, what was a hit is due to noise, or you miss out on hits or on things that could have been hits because your system is not showing the sort of signal definition that you need. So really the opti-ox platform allows for the scalable and reproducible production of large batches of these cells that all do the same thing, as well as the fact that they have the advantages that all the cells are doing the same thing at the same time.
Okay. I think we're coming very close to our end, so I just want to ask one last question. The question is, I would be keen on using these cells. What do I get in the tube? Do I get the cell or the cell lines? Current cells available take weeks to differentiate to neurons.
Yeah, sure. So our cells that we've produced, so they will have undergone an induction phase so that they are primed to already reprogram to the lineage that we're making, and then they're cryopreserved. And so when they're shipped to you, these cells rapidly mature and become post-mitotic, and they are, so the speed to reprogramming and differentiation is much faster for the end user. You also never have to know how to culture an iPS cell. They're ready to use. And really, that's another great advantage of the cells that you'll get in that tube.
Okay. So I think we're just about out of time, and we want to be respectful of everyone's schedule. As a quick reminder, after you leave the webinar, you will be redirected to a brief survey. If you have a moment to spare, we would really appreciate your feedback. Thanks again to Dr. Michael DeAngelo of BitBio. And thanks to all of you for joining us here today. We really do appreciate it. Until next time, remember, science doesn't stop, and neither will we. Stay safe and have a good day.